Hello everyone, welcome to my channel again. Today I'm coming to discuss the hydrolyzable lipids with you, especially the lipoproteins. But before I do that, let's talk about bile salts, which is a biomolecule derived from cholesterol. Now, when we are talking about bile salts are synthesized from cholesterol, like I said earlier, and this is done in the liver. So after the synthesis of these bile salts or bile acids, it is then stored in the gallbladder. Now, when do the gallbladder release this bile salt or this bile acid? The gallbladder releases this bile into the small intestine after an individual has eaten a fatty meal. Now, the function of this bile acid or bile salt is to act as a biological detergent, or it solubilizes fats, or it emulsifies fats, converting these dietary fats into micelles of bile salts. These micelles are actually spherical in shape, which in turn have a dipolar nature. So, example of these bile salts is the cholic acid, the thyrocholic acid, and glycocholic acid, which are polar derivatives of cholesterol. There are two types of bile salts or bile acids. We have the primary bile acids and the secondary bile acids. Now, the primary bile acids are the bile acids that are synthesized directly from cholesterol and it occurs in the liver. Example is the cholic acid. Secondary ones or the secondary bile acids, they are actually synthesized from the primary bile acids in the intestine. So immediately the primary bile acids from the gallbladder has been released into the intestine through the action of bacteria in the guts or the small intestine. A secondary one can be formed. An example of this secondary bile salts is the deoxycholic acid and the litocholic acid. Now, the thyrocholic acid and the glycocholic acid are bile acids that are conjugated to taurine and glycine respectively in the liver. So this is a structure of the cholic acid. You can see it is bearing exactly the same format as cholesterol, just that there is a change at this position. The same applies to this part, which is the thyrocholic acid. It exhibits the same format as the cholesterol, just that there is a change in structure at this point. Now, there are some conditions that we need to consider. This is a gallbladder. Let me see whether I'll see a nice, yes, this is a gallbladder. Now, this gallbladder is associated with a disease called gallstone, which is a calcium stone that is formed inside the gallbladder. And this occurs when the cholesterol level has been elevated above the saturating amount. In chemistry, we learned that there are three types, which is the saturation, unsaturation, and supersaturation. Saturation means it can no longer take in more. So when the cholesterol level is above this saturation level in the gallbladder, or when the cholesterol level is above the saturation level in the bile found in the gallbladder, then this means that automatically some of the cholesterol is going to precipitate out as hard deposits, and this is what we call the gallstones. So as you can see here, this is our gallstone, this is our gallbladder. And as you can see, when the cholesterol is being synthesized, or when the bile is being synthesized from the cholesterol, it moves into these tubes, these tiny tubes, and it is being stored in the gallbladder. That's why when an individual eats a fatty acid, then the gallbladder releases the bile to emulsify these fats. Now, let me go back. So this is the hard deposit that forms when the cholesterol level is very high in the gallbladder. So when these hard deposits are found in the gallbladder mixed with the bile, when an individual eats a fatty meal, this is where the problem comes. The individual will have a severe pain because what's the function of a bile? Is to emulsify the fat. So where the bile is supposed to pass, this narrow tube that the bile is supposed to pass, is being occupied also by what we call the gallstones. And now these gallstones will be constricting this narrow tube. As you can see, the gallstones are even larger than the tube in which it is passing through. So it will cause a very painful condition. Now that is all I wanted to discuss with you about the biomolecules derived from cholesterol, specifically the bile salts. There are a lot of biomolecules that are derived from cholesterol, but I only touched on the bile salts because that's the most important part I want you to know at the moment. Now let's try and move to the hydrolyzable lipids. And as you are talking about hydrolyzable lipids, you know they contain ester bonds, and this makes the molecule available to be hydrolyzed by water or be broken down by water. Let's take our first one to be the lipoprotein. This is lipoprotein. This is the structure of lipoprotein. As you can see here, it is spherical in shape and large, and a complex of lipids. It contains different kinds of lipids. As you can see, lipoproteins are soluble transport forms of lipids in the blood. When you're talking about lipids, all what comes to mind is hydrocarbons. Hydrocarbons, for that matter, they are non-polar. There is this one called lipoprotein. I have already discussed with you. If you hear lipo, something like lipids. Now the protein, which is ending as a suffix, this means that it contains a protein backbone. Proteins are polar molecules, as we all know. So the lipoprotein, examples of the fat or the lipids that the lipoprotein contain is the triacyl glyceride, which is what you are seeing here, the stranded part of it, and cholesterol. The cholesterol are actually free cholesterol, which is found at the outer layer, and we are having our phospholipids. This is a phospholipid. I'm having a clearer image that will make you understand what I meant by phospholipids. And we have our cholesterol ester. This is the cholesterol ester. It is found inside cholesterol ester in a way that the OH group on the cholesterol has been esterified. Yes. So let's move on. You see that this increases their lipid solubility in aqueous blood environment. These molecules or these lipid complexes, it increases their lipid solubility in aqueous blood environment. Now this is phospholipid bilayer. Phospholipid bilayer, in a way, we are having a phospholipid here, another phospholipid, just that they are inverted. The tails are meeting, so it's a bilayer. The inner part is the hydrophobic part, and the outer part is what we are seeing as a polar. So this is a typical example of a phospholipid. We are having our polar head, automatically we are having our non-polar head or hydrophobic tail. Now look at the polar head. From the polar head, we are having the phosphate group here, and we are having our glycerol. The same thing implies here. Now let's talk about the hydrophobic tail. The hydrophobic tail contains only hydrocarbons, and we are seeing a kink here or a bend here because there is a double bond. This double bond, like I explained earlier in my fatty acids, these double bonds actually causes a kink or a bend in the chain. So this is the hydrophobic tail. A similar structure is also applied here. Now let's talk about classifications of lipoprotein. So lipoproteins are classified according to their density, and there is a nice way of classifying these lipoproteins using the electrophoresis of lipoprotein. The electrophoresis of lipoprotein is just a laboratory technique that is used to separate and analyze different types of lipoprotein in the blood based on their size, their density, and electrical charge. Lipoproteins are particles that transport lipids. Like we said earlier, simply they transport lipids or fat through the body. And example include the chylomicrons. It's a very large molecule but least dense. It has the least dense. The very low density lipoprotein, which is the VLDL, low density lipoprotein, which is the LDL, and high density lipoprotein. Now, let's try and talk about how this electrophoresis work. So a sample is just placed on a gel and subjected to an electric current. As you can see, this is the electrophoresis that I'm trying to discuss. Now, different lipoproteins move through the gel at different rates based on their size and charge. Now, this is how the separation occurs. The lipid or the lipoprotein separates into distinct bands on the gel. So these are the bands. So as you can see, we have been separated. We are having our origin here, we are having our chylomicrons. So they are being separated based on their size and also their charge. Now the analysis, the separated lipoproteins are analyzed to determine the relative amount and to assess the risk of cardiovascular diseases. That is all about electrophoresis of lipoprotein. So what are the importance of this electrophoresis that we are doing here? This electrophoresis is actually a technique that helps in understanding the distribution of lipoprotein in the blood and can provide valuable information for diagnosing and managing conditions like the cardiovascular diseases, of which we've talked about and I'll come to talk about again. So now, when you are talking about the chylomicrons are actually assembled in the intestines. That is their original location. They are found in the intestines and usually from dietary triacylglycerol, which is the TAG, and cholesterol. That is where you are going to see your chylomicrons. They contain mostly the TAG, which is the triacylglycerol. Triacylglycerol, I said earlier, they are very non-polar, fully made up of hydrocarbons and are least dense, like I said earlier. So the triacylglycerol is hydrolyzed by action of lipoprotein lipids lining in the blood vessels near cells that uses the triacylglycerol. So the removal of this triacylglycerol results in cholesterol-rich chylomicrons remnants that are taken up by the liver and assemble in the very low density lipoprotein. So let me show you something here. Yes, so the moment you reduces the level of triacylglycerol, what do you think is happening? We are having cholesterol ester, we are also having cholesterol. So the cholesterol level will actually increase. That's all they are saying. Now let's talk about, so the main function of the chylomicrons is to actually transport cholesterol or triacylglyceride to the liver. Now this triacylglyceride or the cholesterol that it is being transferring to the liver or transporting to the liver is not fully synthesized. So it is being transported to the liver to be fully synthesized. Now this very low density lipoprotein, they are assembled in the liver, so they are rather found in the liver. And they contain mainly the triacylglycerol, but this time around the amount of this triacylglycerol is very less as compared to the one in the chylomicrons, and they deliver the synthesized triacylglycerol. Like I said earlier, in quotes, the chylomicrons is transporting the cholesterol, which is not fully synthesized. The moment it gets to the liver, it becomes fully synthesized and it is being transported by the very low density lipoprotein. It transports this synthesized triacylglycerol to adipose tissues and peripheral tissues. Examples of the adipose tissues that we are talking about here, or examples of the peripheral tissues, we are having our adipose tissue, we are having our muscle tissues, and we are having our brain tissues. Also, for the storage of energy, that is the function of the very low density lipoprotein. After delivering the triacylglyceride or the triglyceride, the very low density lipoprotein becomes an intermediate. It becomes an intermediate in a way that it exists between two things, either the very low density lipoprotein and the low density lipoprotein. So that intermediate there is what we call the IDL, which is the intermediate density lipoprotein, which carry cholesterol to the tissues. So So note, it's not the IDL that transports cholesterol to the tissues. It's the very low density lipoprotein. Remember, the intermediate density lipoprotein will eventually be transformed into LDL. So now this LDL, or this very low density lipoprotein, is the one which carry cholesterol to the various tissues. Now, high level of very low density lipoprotein can contribute to the build up of plaque in the arteries, increasing the risk of cardiovascular diseases. Build up of plaque. What are you trying to mean by that? In the arteries, where you find where you find your cholesterol or fats being deposited in the inner surface of these arteries. For that matter, it will minimize the efficient flow of blood through the body system. When this happens, it can lead to a higher risk of cardiovascular disease such as stroke and hypertension and all those stuff. Now let's talk about the low density lipoprotein itself. So it contains mainly cholesterol. This time around, the amount of the T aging is less. It is involved in the regulation of cholesterol metabolism, and the main function is it acts as a major carrier of cholesterol in the blood. So it carries cholesterol throughout the blood, or it carries cholesterol throughout the biological system of humans. Now, the low density lipoprotein is considered as a bad cholesterol. And why is it? Because after the cholesterol that has been used up has been sent to the liver, this low density lipoprotein tends to send these cholesterols back into the bloodstream after circulation. For that matter, it increases the level of cholesterol in the blood. And if you are having the level of cholesterol in the blood, this time around not in the liver, cholesterol in the blood, then you are talking about cardiovascular diseases such as the stroke, because eventually there will be a buildup of a plaque. Now the production, so we can minimize this elevation of the cholesterol level by the body producing low density receptors. What does this low density receptors do? The low density lipoprotein receptors, it actually takes up, or it minimizes the cholesterol level in the bloodstream. For that matter, it also minimizes the risk of an individual getting the cardiovascular diseases. Now, to move on with these receptors can work in many ways. It can bind with the lipid, or it can bind with the cholesterol and its associated lipids and send it to the cell itself, not the bloodstream. It will bind with this cholesterol and its lipid associates and move into the cell. For that matter, in the cell, that is where the breakdown also occurs, the enzymatic breakdown. These same receptors can also act as endocytosis. It can act as an endocytosis process. Endocytosis means it is actually engulfing around it. So it engulfs around this cholesterol and its associated lipids. After engulfing around it, then automatically it breaks it down, minimizing the level of cholesterol in the blood. Let's talk about the high density lipoprotein. The high density lipoprotein is actually termed as the good cholesterol, because that one, it tends to remove the cholesterol from the circulation. So as the low density lipoprotein is doing something bad, the the high density lipoprotein is doing something good. So for that matter, it is considered as a good cholesterol, and it picks up cholesterol from the peripheral tissues such as the brain tissues, the adipose tissue, the muscle tissues, and then it's to the liver. So that's the main function of it. Now, this is the plaque I was talking about. This is actually the buildup of the plaque. So if you are seeing this is actually this part of the artery is very healthy because the pile up of the plaque is no more there. But let's consider this part where there is a pile up of fats, especially cholesterol. Here, it's actually going to minimize the rate at which what red blood cells or even blood will even flow through. When this place becomes very narrow, what do you think is happening? The pressure will increase, and that leads to the cardiovascular disease such as the hypertension, the stroke, and other diseases that are related to the heart. Because we all know that the heart is the one that is pumping the blood. So if the blood pump, whatever you do, the rate at which the heart pumps the blood will remain the same. But if the transport system there's a constriction there, automatically to affect the efficiency of the blood of the heart, and this can lead to a lot of diseases. So this is actually a blood plaque, uh, a plaque that has occurred in the inner surface of an artery, and this condition is usually called the atherosclerosis. Now let's talk about the composition of lipoproteins. So the chylomicrons, like I said, is very large but very low in density. And now look at the aspects. Look at the composition here, the weight composition. The triacylglycerol, like I said earlier, it contains high amounts of the triacylglycerol, which is non-polar. The cholesterol extra, I mean few of them, the free cholesterol, like 1, phospholipids, let's say 9, and protein itself 2. So it makes these chylomicrons not even polar as compared to what you are having here. Now let's consider the very low density lipoprotein. That one, the amount of the triacylglycerol is actually more but lesser than the chylomicrons. The same thing applies here. So as you can see, the the rate at which it becomes polar starts increasing as you move from chylomicrons to a high density lipoprotein. And if you move from the down to the top, the hydrophobic part increases, which is the triacylglyceride. For that matter, it causes the molecule to be non-polar. So moving down to up, the non-polar properties increases, and moving down and moving up to the down, you are having your polar properties to increase. Because looking at the high density lipoprotein, look at the amount of phospholipids, and we all know that phospholipids and protein, they are polar molecules, especially protein. Protein is a polar molecule. It's even a dipolar. Sometimes it can act as a zeta ion, where you are having a negative and a positive charge on different sides but on the same molecule. So we will delve into protein later in the semester or later in the day. So try and keep up with my channel, be updated always, and learn more. Thank you for watching this video, and have a nice day.